Hello and welcome everyone. I'm Glenn Harris, Director of Business Development and Strategy from 908 Devices. I would like to thank you in advance for your time as I discuss the Rebel with you today. Despite the significant advances to improve bioprocess productivities with media optimization and feeding strategies, it is still a challenge to obtain timely and cost-effective insight on critical process parameters and critical quality attributes. Upstream process development and cell cultures labs manage hundreds of bioreactors, microbioreactors, shake flasks, and deep well plates during a typical project. Many samples and vessel volume restrictions can really slow down your ability to get timely and useful measurements. These challenges are not shared during scale-up or in a manufacturing setting where the number of bioreactors is much smaller and the volume of media to sample from is substantially higher. During process development, you have a variety of objectives depending on the project you are working on. If you're looking to optimize the productivity or quality of a biologic, you will often take a minimum of a sample a day from your bioreactors to monitor the extracellular nutrient and metabolite levels. This data helps to inform you about the appropriate changes to a base or feed media that you should replenish or fortify if they are critically low in concentration. Before the start of a process, one can sample the fresh media to ensure that it was mixed and formulated correctly. This analysis extends to ensuring consistency of the fresh media to rule out any lot-to-lot and vendor-to-vendor variations. Viral vector production is a growing need in the industry as there is a capacity shortage to support the growing pipeline of cell and gene therapies. Similar to traditional biologics processes, monitoring the process with daily sampling ensures robust productivities and assists in media optimization when transitioning from adherent to suspension cell lines. Finally, there is rapid growth in cell and regenerative therapy development where monitoring the cell media profile is critical to maximize the expansion and efficacy of the cell therapeutics. In most process development and cell culture labs, general biochemical analyzers are used to run samples at line to provide levels of specific high abundant analytes in the media like glucose, lactate, and ammonia. These systems are straightforward to operate and maintain. Their footprint is usually small enough to fit alongside other process analyzers and bioreactors, and they provide relatively quick answers and sample-to-sample turnaround times to fit into the fast workflows many process development groups are accustomed to. Unfortunately, these systems provide limited information on additional high-value nutrient profiles of amino acids, vitamins, or amines that are common in most cell media formulations. Traditionally, the samples must be sent to an external core laboratory or expensive third-party contract testing lab when any more information is desired. Sending these samples out to a central lab or external contract testing site is neither simple nor fast. After a sample is pulled from a bioreactor, they must be filtered to remove all cells and particulates. The samples are then packaged, labeled, and frozen for cold shipment. If you're lucky, shipping may be as simple as walking those samples down a hall. However, it usually means shipping to another site out of your state or country for analysis. Once the samples arrive, they are defrosted as derivatization reagents are prepared for sample preparation. Sample prep for most chromatographic techniques often involves many wet chemistry steps and additional delays for the derivatization reactions to complete. Once prepared, batches of samples are loaded and run. After all samples are run and completed, raw data files are processed by the testing facility to produce reports. These reports of detected analytes and concentrations are delivered days to weeks after you initially pulled and shipped samples away. The lengthy delay eliminates any possibility for timely interventions based on what happened during a process on a biologically relevant timescale. Our solution for you is what we call the Rebel. The Rebel provides comprehensive at-line, fresh, and spent media analysis for bioprocessing labs. The analysis is label-free, supporting high-throughput media analysis while only requiring approximately 10 microliters of a sample from your bioreactor. This minuscule volume requirement suits those researchers who work with small volume formats like microbioreactors, shake flasks, and micro well plates, along with benchtop and manufacturing scale bioreactors. With a fast turnaround time of approximately 15 minutes, each run simultaneously assays over 30 standard cell media and bioprocess nutrients and metabolites. Simple data reports are generated automatically, freeing you of afternoons tied to juggling proprietary data formats and spreadsheets. All this is delivered at a significant cost savings compared to prohibitively expensive core and third-party testing. The Rebel delivers unambiguous answers where and when you need them. Running a sample alongside your bioreactors on the Rebel is simple. Pull a sample from your bioreactor, spin out your cells and dilute. That's as complicated as it gets. Load it in either a septum cap vial or a 96 well plate and hit start. There is no struggling with settings or tuning. The Rebel already knows which knobs to turn, and data analysis is validated ahead of time with automated calibration. At the end of the run, you will have a full report of the analytes detected and their concentrations. Shown on the bottom is the list of over 30 analytes that the Rebel screens for in a single run. When you run a sample, you're not forced to select just a few analytes that you want to investigate. Instead, the Rebel simultaneously looks for everything. After the analysis, the Rebel delivers a single real-time report. Combine your results from the Rebel with your other at-line biochem and cell analyzers to enable actionable insight. You will shave process development cycles by weeks, if not more. The Rebel reports are delivered in non-proprietary formats like CSV and PDF. Since the Rebel comes 21 CFR Part 11 compliant out of the box, a simple audit log is generated. The Rebel can quickly transfer your reports to an attached USB drive or automatically upload them to an Ethernet connected network drive. This allows you access to your findings remotely and to easily integrate them into your preferred data visualization package, electronic lab notebook, or LIMS platform. Did you ever try to reproduce an experiment and it didn't turn out the same? Or do you sometimes question how a cell line grows better in one vendor's media as opposed to another? Fresh media screening on the Rebel is a straightforward application, and it gives you the insight that you've been missing. One of the most used cell media types is DMEM F12. DMEM F12's formulation is well known, and it can be purchased from numerous vendors. We wanted to take a glimpse into the consistency of DMEM F12's media across several providers. The values were normalized to vendor-supplied formulations. As shown here, consistency between vendors should not be assumed. In some cases, specific media components were as high as 2.5 times the expected values. In contrast, other components, like glutamic acid in vendors 2, 3, and 4's media, were not detected at all. When ordering large amounts of cell media, a researcher often decides between pre-mixed liquid and dry formulations. Dry formulations afford the advantages of easier storage, shipment, and costs. However, dry formulations must be mixed ahead of time. This may introduce variation to the composition based on how well directions are followed and the environmental conditions while in storage. The values reported here on the bottom are from the analysis of medium 199 normalized to the specification sheets. Both leucine and phenylalanine were the most consistent amino acids. They both deviated less than plus minus 10% of the formulation value across all 4 medias tested. On the other hand, arginine, lysine, and tryptophan all exhibited variations greater than plus minus 20%. Researchers need to work with established cell media providers to ensure that their cell media choice is appropriate for their application and that the handling of the media once it's received is correct. Researchers should consider a frequent analysis of their cell media whenever new batches and lots are received, periodically during storage, and during experiments. For many fermentation processes, you may rely on complex and undefined media consisting of hydrosylates, peptones, yeastylates, and other mixtures. As a result, these media blends may be highly inconsistent between vendors and lots. Shown here are 3 examples of that between LB, TB, and SOB medias. These are all undefined media types following a known recipe for formulation. But the ingredients to the recipe are all undefined. So depending on where the raw materials are sourced and stored, you might encounter variations. I will highlight just the one instance on the left showing LB media. Vendors 2, 3, and 4 all have similar amino acid concentrations. However, vendor 1 had almost 2 times higher lysine levels and over 360% higher concentration of threonine. An extreme deviation like this would likely cause unexpected process changes. When culturing advanced therapies derived from T cells, early experiments typically adopt the traditional media platform of RPMI media supplemented with 10% FBS. However, there is a growing desire to use chemically defined media that excludes serum and other animal-derived components. The adaptation of a cell line from serum-containing media to a fully chemically defined media is challenging though, especially when no insight into the media is known ahead of time. On the left, we compared serum supplemented media with 3 vendors' media options for T cells. There are many options available on the market today, and they range from complete all-in-one media options to media that should be used exclusively only for early culturing or late expansion of T cells. Let us look at one component, for instance, arginine. We identified that the conventional RPMI media with serum had anywhere between 2 to 3 times more arginine than all the chemically defined media types we tested in this study. This is exceptionally relevant for T cells, since various research teams have identified the importance of arginine for growth and efficacy. I have highlighted a quote below that summarizes these advantages, since it can play an essential role in both in vitro and in vivo studies. HEK293 cells have been a workhorse cell line in cell biology and bioprocessing labs. HEK cell lines are commonly used to produce both therapeutic proteins and viral vectors. Originally, HEK was derived from adherent cells that prefer a compatible surface for growth, but this had limited the platform's scalability. Therefore, there's been a desire to improve both process productivities and consistencies of the adherent HEK cells grown in serum supplemented media. Processes have been adapted to cultivate HEK cells in suspension and with serum-free, chemically defined cell media. But as you can see in this figure, not all chemically defined media for HEK is the same. For instance, the dipeptide alinoglutamine was only detected in vendor 2, whereas hydroxyproline was only detected in vendor 3. Asparagine was 25 times higher in vendor 3's media compared to the next highest composition. In fact, vendor 3 had the highest concentration of roughly 2/3 of all the study's detected components. In contrast, vendor 4 had the lowest concentration in over half of its components. Selecting the right media for your HEK cells is particularly important to produce viral vectors. On the right-hand side of the slide, you can see how both lentiviral and AAV production from HEK cells differs based on the commercial media used. In the case of the lentiviral process, there was a doubling of vectors produced within 72 hours post-transfection by using a chemically defined media. The AAV process made over 30% more vectors using the same optimized media. Implementing the REBEL to support efforts like this would ensure that the new media used is consistent batch to batch, and that you would have the data to support continued customization of the media to boost your titers higher. Now, let's wrap up our discussion of the REBEL with a spent media analysis case study. Automated microbioreactor systems have decreased the complexity of process development experiments. However, the bioreactor's small volumes, typically less than 250 milliliters, limit the media volume one can extract for daily sampling. In this study, we work with a phenomenal team at Clemson University who has an AMBER 250. Together, we describe the means to analyze spent media taken from an AMBER 250 culturing an IgG1 producing CHO-K1 cell line stressed by various levels of ammonia. In this experiment, 2 bioreactors were run as controls. 2 were started with 10 millimolar ammonium chloride added, and 2 started with 30 millimolar ammonia chloride added. Samples were pulled daily in this fed batch run. Each sample was allocated out to a Beckman Vicell, a Roche CDEX, and run on a REBEL. Although it was easy to discern the highly stressed 30 millimolar ammonia culture from the others, there were negligible differences in the standard cell growth and metabolite analytics from the controlled and 10 millimolar ammonia stress bioreactors. Despite this, there is a dramatic change in the amount of IgG produced, shown in the figure in the bottom right. To explain this, we wondered if there was something other than the ammonia changing in the system that could contribute to the observed productivity differences. Here's a snapshot of all the samples from the 6 bioreactors run on the Rebel. Each color map corresponds to a single bioreactor. The color scale is normalized to each media component's initial levels at the beginning of the process. Across all bioreactors, there is a buildup of most essential amino acids throughout the process. On the other hand, relative levels of the non-essential amino acids were more varied. There's a lot of data here to digest. Let's simplify it for this discussion. Here is just a single control and a single 10 millimolar stress bioreactor. We quickly see that while some analyte profiles over the run are similar, like glycine in the light blue, others are very dynamic, like alanine in yellow. Note how the alanine profile for the control reactor on the left goes up, then down, then up again. Whereas for the 10 millimolar stress bioreactor, it goes up and stays high. Discovering these differences is vital in providing clues that explain why productivities may differ between cultures. This may lead to adjustments to the feeding strategy, which could correct the problem in the future. Improving your media selection and assisting in deeper metabolic and process understanding are just 2 aspects of how the Rebel can help you. Fresh and spent media analysis can be used for many applications, like finding out the culprits of sequence variants in a recombinant protein, enhancing cell-specific productivities in real time, creating models that predict process performance, or fundamental studies on cell regulation. We are sure that if you are growing cells, the Rebel can support your efforts. Please let us know if you have any additional questions or inquiries. We have other product literature and videos available on our webpage. I would be very happy to assist you in getting a Rebel or 2 into your labs this year.